Thank you very much. First of all, thanks very much for the acknowledgement. I'm lucky to be able to also say that we now finally have a permanent director of the Office of Vaccines in place as of yesterday. So I will at least be able to step back a little bit from oversight directly of the vaccine day-to-day work. So breathing a sigh of relief, although I'm very grateful that you're allowing me to participate virtually. We had some regulatory actions this morning that had to be taken care of. For those of you who may be interested, we've authorized boosters for the mRNA vaccines down to the age of 5. So those are the bivalent boosters, updated ones. So more to come there, but that's not the purpose of this meeting. So what I wanted to talk to you about are some of the efforts that we are doing to try to help take cell and gene therapy to the next level. I'm the first to acknowledge that over the past years, we've had good intentions to try to get to places, and we've been slowed down, some by the pandemic, some by forces of nature, other forces of nature. But I think we really want to try to see what we can do to move things forward here. And just to kind of give you an outline here, want to currently just overview where we stand. You probably are all very aware of that, but just our current state of cell and gene therapy, talk about overcoming barriers in development, talk about the case for global regulatory convergence, which increasingly to me seems like something we have to have more of a discussion about, and talk about some center updates. So just where we stand right now, we are now up to 10 approved gene therapies. These are both cell-based and directly administered gene therapies in the United States. Although the majority right now are CAR T cells, we now have cell-based gene therapies on this list, and directly administered gene therapies. And the future is bright. There is a robust global pipeline of products. I think we are seeing in the genetically modified T cell space for cancers, more and more complex products coming along. We're starting to see allogeneic products make their way through. And with the use of CRISPR-Cas9 and other genome editing technologies, starting to see chimeric antigen receptor T cells that might actually address solid tumors because of the ability to engineer multiple receptor recognition sites. Obviously, hemoglobinopathies, hemophilia A and B, lysosomal storage disorders, neuromuscular diseases, retinal disorders, and others. In fact, I think it's really an exciting range of disorders, but there are some challenges, and that's what I think we need to try to address. Just to show the success, I think we often point to anasemnogene or Zolgensma, which is for a relatively more uncommon disease, spinal muscular atrophy type 1, which is really a terrible disease that leads to a loss of motor function early in life. This generally leads to children dying of complications by age about 3. And with the introduction of this gene therapy, really has transformed this in a simple manner, rather than having to have potentially repeated injections of an oligonucleotide. This has led to a once and done intravenous treatment that has really led to remarkable responses with children now developing normally, and the child on the right evidencing that a child running around with spinal muscular atrophy type 1 at age 3 that's just not possible without something like this intervention, and that is why I think this is just an example where when this goes right, it's quite remarkable. and some might describe it as miraculous. We may not believe in miracles, but there are things that are miraculous, and this is one. And these gene therapies for disorders that are very rare are in aggregate really important, because when you take all of these diseases and put them together, they affect quite a number of people. But more so than that, as we move into the era of molecularly targeted therapies where we're actually targeting the genome with correction, we may need to have paradigms where we're going to be correcting the base pair in that will only be present, that correction may only be needed in 20 or 30 individuals. So getting this paradigm for how you make gene therapies that touch a small number of people right is important. Additionally, I still would say that if we want to get large-scale gene therapy right for lots and lots of people, this is the correct place to start, where the need is incredibly great, where we can potentially look at a variety of different applications and learn a lot. And really, the therapies that we're talking about now are in this space of individualized medicine, where we really are creating the right drug to treat the patient. The chimeric antigen receptor T cells that are autologous are really an example of customized products. There are others in development now, such as personalized cancer vaccines. They've been in development, some of them in development for quite some time, but there are customized products. And then there are created products like these gene therapies. Our goal is to try to help streamline the creation of important products for patients. But what are the barriers in development that need to be overcome? Rather than overviewing everything, I figured we would talk about both manufacturing and product access. And when I'll talk about product access, I'm talking about from the FDA's perspective. So I'm not going to be talking about it from the payer perspective. So this is a slide I often show, which basically shows that commercial viability in 2022 has not changed much from the past, which is that we still have a sweet spot in the middle here of making gene therapies for the hundreds to few hundreds of patients. The very large scale, production of gene therapies is something that's is a story for another day. As we learn to have better vector production methodologies, and we feel comfortable with the evidence. The problem really is in the very small scale area where we have to find a way towards commercial viability if we're going to succeed, and we have to figure out what's going to get us there. Some would argue that, well, this is just something that should be handled by philanthropic groups. But I would put it to you that we're not going to find enough philanthropic groups to foot the bill for these gene therapies for the hundreds upon hundreds of different diseases that need to be addressed. We're going to have to find a way to make this commercially viable so that industry can find a way forward towards this. Now, one way of getting there may be to change how we think about making these things. It turns out that for certain things like AAV production, you can actually automate a fair amount of these things. And we are in the process of working with various collaborators to try to see how much of this can be automated. This is made possible by any number of things, including the fact that we now have various sensing technologies. You can put the right controls in place into systems. But ultimately, I'm not going to bore everyone with manufacturing steps, but the goal would be to get to something that is a device. And I know it's a soda dispenser, not a gene therapy manufacturing device, but it's a stand-in for what might help here, which is if one could spend most of the time worrying about the actual constructs that one was generating and less time about the manufacturing, including the purification and formulation of the product, we might be able to see this reach commercial viability more rapidly. And so some of the other things we've been talking about, but we're finally making some progress on, is getting towards putting together a cookbook type approach for the development and manufacturing of bespoke therapeutics. We do believe that if academics in particular, where a lot of the innovation comes from, could follow the same playbook more of the time, it would be easier for them to transfer work to contract manufacturing organizations without having to rework everything. That would be mutually beneficial. And it would likely add value on both ends of the equation to the investigators who are potentially licensing out technologies, as well as to those who are recipients who would like to have more surety that the technology will actually transfer smoothly. And then a piece that we're working on in FDA is to look at how we can use our regulations to allow us to leverage non-clinical and manufacturing data from one application to another. So initially, our feeling is that we'd start with allowing an individual company to leverage the information from one application to another. And then at some point, if that's really working well, we can consider expanding that concept further. The idea here is that really there are certain pieces of gene therapies that are not like your typical small molecule drug because they're reused repeatedly, and provided you don't either understuff them or overstuff them, you can probably get a similar result when putting a similar type of insert in other words, an insert that produces a similar type of either secreted protein or membrane protein, et cetera. And the idea here is that if we could get this paradigm to work, rather than having a manufacturer go back and do all of the preclinical tox information, give us all the manufacturing information each time when they are submitting something, they would basically just cross-reference. And that would, again, allow us to focus on innovation that's going to bring benefit to people rather than having a lot of what used to be paper. We used to kill trees. Now it's basically using energy for storage in data. So it still kills trees, basically. The idea here is, though, to allow us to get innovation to those in need more rapidly. How can we, get access here? Now, our traditional access is either through investigational products, through investigation new drug applications, through either research INDs, the traditional INDs, or through expanded access programs. And then products hopefully move on to get either a traditional or an accelerated approval. I want to say a few words about each of these because we are going to try to lean into accelerated approval as much as we can. I also put in here emergency use authorization, but that really would be for a very interesting situation in a public health emergency setting. I don't have listed here right to try because in general, most gene therapies that we're talking about today are not going to have a phase 1 study completed that might allow one to do right to try. And there are other considerations as well that make that potentially suboptimal. But for expanded access, this is really a program for serious diseases or conditions when people can't end up being treated on clinical trials. The benefit has to justify the potential risks. And obviously companies have to be willing to make it available. It's potentially a good complement sometimes to more traditional clinical development programs when there are individuals who might not fit into the clinical development program. And I think in the gene therapy space, the one thing I would just emphasize is that we very much understand that those individuals who might be treated on an expanded access program with a gene therapy who have serious diseases, they may be different enough from those who are being treated in the protocol that adverse events that might occur with those on an expanded access program don't count against you in the traditional development plan. Now, I don't want to say that 100% of the time, because obviously, if there were very serious events that started to be seen in all of the patients, that would be one story. But what I'm just trying to say is there's always been a hesitancy for expanded access, or oftentimes there's a hesitancy for expanded access programs because of that concern. And I think we've come to acknowledge that those populations may be different. Then there's the issue of what we have with our BLAs. Oh, by the way, just to say, individual patient access is something that we are facilitating through single patient IND requests. There's a pretty simplified form, and the bar for this is reasonably low. We have intermediate size population access, which is where most sponsors decide to go because it can be run under a protocol, and it fits once there's some evidence that a product has effect. But our biologics license applications are what most people are aiming to get, where products have to be demonstrated to be safe, pure, potent, and effective. And we have to see substantial evidence of efficacy from adequate and well-controlled trials. The issue here is what do you end up with? Well, a full approval is when you have a clinical endpoint, such as how patients feel, function, or survive. But we recognize increasingly that for gene therapies, we may at times need to be relying on our accelerated approval authorities, where we're looking at either intermediate endpoints or surrogate endpoints. And that's because, particularly as we move on to some of the solely progressive diseases, diseases that still really incredibly impair people's quality of life over the course of years, in order to do a clinical trial timely, one will have to look at some biomarker or intermediate endpoint in order to get the products across the finish line to an approved product that can then be looked at over the course of time by either longer clinical studies or real world evidence to convert them to full approval. And so I think stay tuned as we say more about this over the coming months and years. I want to just, in the last 5 to 10 minutes here, just talk a little bit about how we try to do this better globally, because it has not escaped our attention at FDA that there have been some clouds on the horizon in gene therapy. Gene therapies that were launched, for instance, came off of the market in certain locations, or gene therapies that were licensed to companies where the companies returned the assets back to the original academic investigators. And we realize that there's some of this that needs to be addressed with what we can address with our regulatory environment in the United States. But some of this also may relate to how we can make gene therapies for small populations more widely available, because what may be a tiny population in the United States becomes a reasonable size population when you go globally. For instance, the difference between 30 patients and 100 patients may make the difference between not commercial viability and commercial viability. And so how do we get there? Well, we know roughly that commercial viability is about 100 to 200 gene therapy treatments a year. And as I've already said, one country may not have this, but we could potentially think about ways we could get there. by finding ways in which we could facilitate gene therapy introduction into multiple high-income countries at a given time, and then potentially adoption into low and middle income countries. And, I say this because for certain diseases, particularly in the area where I have some training and expertise in hematology, where hemophilia, hemoglobinopathies, it's individuals in low and middle income countries that may stand to benefit the most from gene therapies, because the current standard of care for individuals with these disorders is really not good. And many individuals die in childhood, or they live very challenging lives because of the lack of access to supportive care, whereas gene therapies could transform that without this constant need for supportive care. But that would rely on providing a regulatory framework that would need to be somewhat harmonized, and perhaps we'd need to have something like pre-qualification, an equivalent for gene therapies for the regulators. And that's because many of these countries don't have experience in the regulation of cell or gene therapies. But this could potentially make a big difference in access for, globally, make a big difference to patients. And this concept of global regulatory convergence might make it possible that it's, at least starting in this rare disease space, might allow us to get these therapies to more people. Again, networks in high-income countries would be helpful, and then that might also be able to spread to low and middle income countries. I think the other piece of this is that we really need to understand that we do have to have global regulatory collaboration, because if there are bad actors in the gene therapy field, it will create problems throughout the ecosystem. I think we already all know that. There are a bunch of areas which we can potentially converge. And right now, I think I hear routinely from companies that are operating in multiple regulatory jurisdictions, the problems that there are different preclinical study requirements, especially for tox studies. The environmental assessments are different from place to place. The amount of manufacturing information required is different. I know that firsthand from some of our experience and the experience of our sponsors between even EMA and FDA, and we think we're pretty close together. And sometimes there are different clinical outcome requirements. So hopefully, if we could converge here on these, especially around these very small populations, we could make a difference for people. We are hoping to start to get a convergence of regulatory approach in high-income countries. We're trying to eventually get to harmonization, but that might take a while. There is an effort underway through WHO to lead to general convergence in how we discuss cell, tissue, and gene therapies. And that's the idea, is trying to provide the education needed in low, middle income countries to prepare the way for potentially using gene therapies. But hopefully, with meetings between high-income country regulators, will start to converge. And that could make a difference. In the meantime, while that's going on, we'd really encourage sponsors considering global development programs to invite regulators to early stage meetings. At least from the standpoint of FDA, we have interact meetings, pre-IND meetings. We always, if the sponsor would like to have a global regulator present and they do the inviting, we will not, we'll always invite them and be happy to have them there. So we have this white paper that we're working on with WHO, and we will continue to try to move ahead, advancing methods for production of gene therapies and facilitate public-private partnerships. But finally, I'd like to just tell you some center updates. People probably have heard about a reorganization that has occurred. It is a second reorganization, but the last reorganization that we did in the Office of Tissues and Advanced Therapies was about 3 years overdue. So this was one that was undertaken to adjust to what has become very necessary, which is we basically have an office that is grown in leaps and bounds. It's going to, with the user fee resources that will be coming, we'll be starting to get towards the 500 mark in terms of individuals, and the size of the office was becoming very difficult to manage. In addition, the ability to deploy the specialty experience within the office was also challenging. So with this reorganization of the Office of Tissues and Advanced Therapies into the Office of Therapeutic Products, we will have essentially an office devoted to the manufacturing of gene therapies, an office devoted to the manufacturing of cell therapies, a large clinical review branch that will be able to deploy clinical reviewers across where they're needed, pharmacology, pharm tox experts, and so we're thinking that this will, and we'll also have, by the way, I neglected to mention a group focusing on our more traditional plasma therapeutics. So all of that, I think, will help us allow us to increase the interactions that we need to with various stakeholders, both patients and industry. That will, most importantly, I think for me, is to improve the timeliness of our responses and our meetings, which I really think we want to make a big improvement in over the coming years, and also enhance the consistency of our responses by having offices really focused, more focused, because again, feedback we hear from industry loud and clear is that is something that we would like to have. And of course, we'd like to have that as well. Just finally, as we, we will over the next, whether it's going to be, some of these are shorter term goals, some of these are longer term goals, but we certainly want to work to more clearly define the use of accelerated approval for gene therapy. And we may do that in various ways, up to including consideration of guidance in this area. We will work to more clearly define development pathways for cellular therapies. I think that is something that, I think at the moment, there are a lot of different cellular therapies outside of gene modified or genetically modified cell therapies, and we want to help make those development pathways more clear. And very much we want to advance manufacturing technologies for cell and gene therapy, because I think that is one of the keys to bringing down cost of goods and hopefully making them more available. So all in all, we're very committed to cell and gene therapy development, and we I think we want to both make sure that first we get it right with smaller populations, but then hopefully see this grow to be able to bring the benefits of cell and gene therapy to larger populations. And we'll use all the tools in our toolbox to help make that happen. So thank you for listening, and look forward to the next panel. Thank you.